Well, welcome everyone to this director's lecture. I'm going to start by acknowledging the traditional owners on the land on which I'm sitting, and I suspect many of you are sitting, the Wurundjeri people of the Kulin Nation. I pay my respect to their elders. I'm absolutely delighted that Tom Kay and Helen Thomas are coming back to WeHi for an hour or so to let us know about their work over the last decade or so to characterise inflammatory pathways that lead to beta cell destruction in type 1 diabetes, and how that's inspired a clinical trial that's about to get underway in Melbourne to test whether an inhibitor of interferon gamma signalling, JAK inhibitors, can preserve beta cell function in type 1 diabetes. Tom, I was not at the Institute when Tom was here, or when Helen was here for that matter. They've both been here in the 90s and the early 2000s before moving across to St Vincent's Institute. Tom is now the director of St Vincent's Institute, and Helen is a lab leader in the diabetes and immunology unit there. And they've worked really as a pretty powerful team, initially in NOD mouse models, followed by some islet work and pancreatic islet transplantation, and most recently in this very interesting story that Tom will kick off with and Helen will follow up with. So I hope you enjoy it, and we'll save some questions for the end. Thanks very much, Tom.
Thanks a lot, John. Can you hear me okay?
That's great.
So thank you very much for the warm welcome, John, and thanks everybody for tuning in on this beautiful Melbourne spring day, which we can all see in the background at John's house there. And I hope everyone's managing okay in these very difficult times. As John said, look, both Helen and I worked at WeHi in the past. I actually first worked at WeHi in 1978, which I'll say seems several lifetimes ago, but it's always a real pleasure to come back and give a talk. And we're going to talk, as John said, about our upcoming clinical trial. Now, hopefully my slides will advance. Maybe I need to. There we go. I'm not sure they're going to advance. Oh, yeah, there they go. They are going to advance. Okay, so I'm going to tell you a bit about the background of type 1 diabetes, and it's a very highly cell-specific autoimmune disease. So really only the insulin-producing pancreatic beta cells are destroyed in this disease, not the neighbouring, very closely related cells which produce other islet hormones. And the pancreatic beta cells are destroyed by the immune system, specifically by autoreactive cytotoxic T cells, and that results in insulin deficiency and the inability to control blood glucose, as we all know. And the genetics of the disease, which we're not going to go into today, are dominated by the HLA and other immune system genes, the HLA being the MHC genes in humans, and there are also environmental triggers in early childhood that are poorly understood and being very actively investigated, including at WeHi by Len and John and their teams and their collaborators. So our goal is to develop treatments for diabetes that target mechanisms of disease, especially immune mechanisms, and then test these in clinical trials. Diabetes is currently treated by hormone replacement therapy with lifelong exogenous insulin, and of course the disease mainly starts in childhood and adolescence. And amazingly enough, insulin's been the treatment for diabetes for nearly 100 years, since Banting and Best purified calf insulin and injected it into Leonard Thompson in 1922. So it's interesting to think whether any other major treatments for important diseases have lasted that long. And the hormone replacement treatment with hormone replacement therapy with insulin, now very closely linked to blood glucose measurement, is increasingly sophisticated, but it's still very burdensome to balance insulin, glucose intake through food, blood glucose, exercise, and other factors. And not surprisingly, many patients, especially adolescents, don't reach treatment targets, and that increases the risk of long-term complications of the disease, particularly renal impairment, kidney disease, visual impairment, neuropathy and so on, and reduced life expectancy, particularly due to cardiovascular complications. So, as I said, our goal is to test new treatments for type 1 diabetes in clinical trials. And there are two main types of clinical trials that are done in type 1 diabetes, prevention trials and new onset trials. The left panel explains the concept that children develop autoantibodies, and let's just turn on the pointer here. That children develop autoantibodies early in childhood. A median age of onset of autoantibodies at about 2 years of age, and then they progress to the diagnosis of diabetes. Depending on how many autoantibodies are present, they progress to the diagnosis of diabetes over a period of years in most cases. So an intervention trial in these stages of diabetes before hyperglycemia has developed, it's got the outcome of preventing or delaying progression to diabetes. So that's a prevention study. On the right is the outcome for intervention trials done after the diagnosis of diabetes, which is the kind of trial that we're going to be doing and we're going to be discussing today. An intervention at diagnosis, some endogenous insulin is still produced, and this declines over the first 12 months and onwards after diagnosis. And an intervention is tested to see if it can reduce this rate of decline in endogenous insulin production. And that's measured by how much C-peptide is detected in the blood after a stimulus to insulin secretion, such as a standard meal. C-peptide, of course, is part of the proinsulin molecule that's removed when mature insulin is produced, and it's co-secreted from the beta cell with insulin. Now, clearly, in many ways, having diabetes or not, this prevention study format is a better endpoint for a clinical trial, but these trials are extremely difficult to do because of problems of finding autoantibody positive subjects. And it's been a tremendous achievement of the WEHI group led by Len and John and their collaborators to do 2 of these trials in recent years. Intervention trials of both types have been carried out around the world. Many more new onset trials than prevention trials. And some of these trials are shown on this slide. Only a fraction of the trials are shown on this slide, and it's a complex slide. So on the left of the slide, in the background is this model of how type 1 diabetes develops, with beta cell mass gradually being lost after the onset of autoimmunity. And this period before clinical diagnosis, which we used to call preclinical diabetes, is now called stage 1 and stage 2 diabetes, and after clinical diagnosis is now called stage 3 diabetes. This shows some of the agents that have been used to intervene in both newly diagnosed stage 3 or before that in stage 1 and stage 2. Now, teplizumab, shown here, which we've highlighted on this side of the slide, and the outcome of that recent study using teplizumab in stage 2, a prevention trial of diabetes, is perhaps the latest, greatest news in the field. And teplizumab is a form of the OKT3 monoclonal antibody, which was used in the 1980s to treat organ transplant rejection. And it was modified by Jeff Bluestone many years ago to reduce the cytokine storm that was a feature of OKT3 therapy. And this data shows that a single course of teplizumab in stage 2 can delay the onset of type 1 diabetes by about 2 years. Now, none of the treatments on this slide have been approved for clinical use by regulators, whereas many of the drugs here are in use for other autoimmune diseases, particularly rheumatological diseases, but also other diseases. And the reasons they haven't been approved in diabetes are several. but they include the fact that there's a high bar to introduce new therapies for a disease that's primarily diagnosed in childhood, for which there's an existing, albeit imperfect and burdensome treatment, and because of the concern about substituting these drugs for insulin and the possibility that you're just substituting one set of side effects for another. And there's particular concern about immunosuppressant drugs that impair immune responses to tumours and infections. Now, as it said in the title of the seminar, the data we're going to show today will be a mixture of studies in humans with type 1 diabetes and an animal model called the non-obese diabetic mouse model. And this is necessary because it's unusual to have access to the pancreas in humans with new onset diabetes when the active phase of the disease is going on. And of course, because the intervention studies you can do in humans are quite limited. Now, like all animal models, the NOD mouse is not an exact copy of the human disease. And one major difference is shown in this part of the slide, where it shows that in the NOD mouse, more female mice than male mice develop diabetes, whereas this is not the case in humans. The numbers are close to balanced in humans, or perhaps slightly more males. But some of the similarities is that it is a spontaneous autoimmune disease in the mouse. It begins with genetic and environmental factors which overlap with some of the particular genetic factors in human diabetes, and then what follows is immune invasion of the pancreas, increasing insulitis and beta cell destruction, and this really, if you like, similar to stage 1 and stage 2 diabetes, and then development of hyperglycemia. And down here is shown the pathological appearance of the immune cell invasion of the pancreas. Now, of course, we're not the least bit interested really in the end in curing diabetes in mice. We're interested in curing diabetes in humans, but the mouse model is a useful clinical stepping stone. Now, I'm just going to talk a bit more about the background of the particular project that we are doing. So we're interested in particularly in CD8 positive T cells, cytotoxic T cells, and their role in the development of diabetes. And we know from studies in human diabetes that they are, and this is some immunohistology from a pancreas that we studied in our laboratory from a young man who unfortunately died of type 1 diabetes when he was a university student soon after he developed the disease. We know that CD8 T cells and CD4 T cells are in the islet infiltrate. This is a fancier slide showing the same sort of thing, and in this study that our group participated in showed that some of these T cells in the islets recognise well-known diabetes autoantigens, and there are in fact more CD8 positive T cells in islets at diagnosis than CD4 T cells, and this is some work again from the pancreas that we studied in our lab. And we're particularly interested in understanding how these CD8 T cells recognise and kill beta cells, and they do that, of course, via their interaction with the MHC class 1 protein on the surface of the pancreatic beta cell, presenting islet antigens in the antigen binding groove of the MHC class 1 molecule. And they can then potentially destroy the pancreatic beta cell, and that's going to be, I'm going to show you that in this video that was made by Parag Trivedi during his PhD with the help of Misty Jenkins, who was then at Peter Mac. And in this video that you'll see that the T cells are labelled with a calcium fluorophore, and these T cells recognise an autoantigen in the pancreatic beta cell, and they'll flash green when they recognise the antigen. And there's propylium iodide in the medium, which will be taken up and make the beta cell go red when it's killed by the T cell. And you'll see, you can see the T cell recognising the beta cell and flashing green, and then the beta cell dying and propylium iodide entering the cell. So really, in a nutshell, this is exactly the process that we are keen to block, and we're focused for many years on the regulation of MHC class 1 expression on beta cells, as well as how CD8 positive T cells kill beta cells. Now, in this slide, you'll see in this immunofluorescence panel, and this comes from the work of Sarah Richardson in the UK, and it's studying some islet biopsies that were taken from new onset type 1 diabetes patients by some investigators in Norway. We've done similar studies in our lab, but what you can see here is in the type 1 diabetes islet biopsy, you can see increased expression of HLA-ABC, the MHC class 1 proteins on the insulin-containing cells within the islet. And there's also upregulation of the STAT1 molecule, the signalling molecule that's responsible for stimulating the expression of the HLA class 1 genes. And I think I've lost my pointer. Oh no, there we are. I see. Need to get rid of that. Okay, so we're back in action here, and similar work was done in our lab by Pete Campbell during his PhD, looking at increased MHC class 1 expression on that pancreas that we received in our lab, and we know that in vitro cytokines, including interferon gamma, can upregulate the expression of MHC class 1. And in this study over here from the mouse model shows the increase, that interferon gamma is indeed expressed in the islets of nod mice as insulitis progresses through the course of diabetes in the mouse. And this very important historical slide here shows some work that Helen Thomas, my colleague, will be talking soon, did during her PhD, where she blocked the impact of interferon gamma on the pancreatic beta cell using a transgenic dominant negative interferon gamma receptor. And you'll see here that in the non-transgenic mice, there's increased expression of class 1 MHC on the beta cells, and that's blocked. When interferon gamma action is blocked, the class 1 expression goes back down to the baseline level seen in nod skid mice that do not develop any immune infiltration, of course, of their islets. So we're very interested in exploring whether this reduction of MHC class 1 expression, whether the reduction in the impact of interferon gamma would also protect the mice from diabetes. And we did this with a different transgene. We used here transgenic nod mice that expressed the molecule suppressor of cytokine signaling 1 in their pancreatic beta cells. And this work was done by Mark Chong during his PhD, of course, using SOX1, which was discovered around that time by Robin Starr, Doug Hilton, Warren Alexander, and their colleagues at the WEHI. And you'll see in this panel that class 1 expression on the pancreatic beta cells are shown here, detected by flow cytometry by autofluorescence, that the levels of class 1 expression on the beta cells was much lower than the levels of class 1 beta cells in the non-transgenic littermates. So by blocking interferon gamma action on the beta cells, class 1 MHC levels stayed low. And in fact, in this particular model of diabetes in the nod mouse, that also very much protected the mice from beta cell destruction and the development of diabetes compared with their non-transgenic littermates. So just to summarise where we've got to at this point, what I've told you about is that the immune system secretes, T cells secrete interferon gamma, and that acts via the JAK-STAT pathway to increase MHC class 1 expression. And then the CD8 positive T cells that are autoreactive recognise antigen expressed by MHC class 1. And then they deliver the contents of the cytotoxic granules, perforin and granzymes, to the beta cell to cause apoptosis. And in some work from David Kagi, showed a long time ago that perforin deficiency, the inability to do this, protected the NOD mouse from diabetes. So the data that I've shown you suggests that if a therapy could exactly mimic transgenic expression of SOX1 in beta cells that we've shown in this animal model, if we could do that with a therapy in humans, then beta cells could potentially be protected from the immune system without directly suppressing the immune system itself. And now Helen Thomas is going to explain how we took a version of this idea forward over the next several years, but with several twists along the way. And at this point, I'm going to hand over to Helen, hopefully, and hopefully that technology will work. Thanks for listening.
Okay, so I think I have to, anything you share will replace it. Okay, I'm going to share my screen. Down here. Okay, can you see my screen now?
Yes, that looks good.
Good, okay, so I'll just get my pointer up as well. Okay, so yeah, as Tom said, overexpression of SOX1 in beta cells prevents diabetes. And so this is something that we can test in humans. But as yet, there's no way to specifically overexpress SOX1 in beta cells. But as we know, inhibitors of this pathway exist. And I've taken this diagram from Jeff Babon's 2018 paper showing the way that SOX1 inhibits the activity of the JAK molecules. And although it inhibits the activity with a different action, the JAK inhibitors occupy the binding cleft, the ATP binding cleft of the JAK molecule. And this inhibits its activity in a remarkably similar way to that of SOX1. The diagram left here is a network analysis that we did in our lab of knockout mice made in the NOD model. So over the years, many genes have been knocked out in NOD mice, and these genes that are shown here are all the ones that resulted in at least 80% protection from disease compared with the wild type controls in the same study. So we grouped these genes according to the cell type that they're expressed in. So we've got beta cells, CD4 and CD8 T cells, and then made these networks. And what you can see is the STAT1 gene is central to the pathways that are important for diabetes in the model. So we've known for some time now that STAT1 deficient NOD mice are protected from diabetes, and that's shown here in this paper from Myung-Shik Lee's lab in Korea. And that JAKs are direct activators of the STAT proteins downstream of cytokines, and I've just put this little diagram here to remind us of the signalling pathway. So there are, so SOX1 is a negative regulator of the JAK-STAT pathway, and there are many cytokines that use this pathway in their signal transduction. These include the interferons that we've talked about already, and also common gamma chain cytokines, which are important for lymphocyte proliferation and homeostasis, the IL-6 family, and some blood cell development cytokines such as GM-CSF. So JAK inhibitors will block signalling by these cytokines, and depending on the selectivity of the inhibitor, of course. So many JAK inhibitors have been developed, and this slide just lists the JAK1 and JAK2 inhibitors that are on the market, including the JAK1/2 inhibitors ruxolitinib and baricitinib. Baricitinib is marketed for rheumatoid arthritis, and the JAK1 selective inhibitor upadacitinib, which is marketed by AbbVie for rheumatoid arthritis. So these drugs have been approved for some diseases, including rheumatoid arthritis, inflammatory bowel disease, and psoriatic arthritis. And They're in trials for many other diseases, including some in children, such as juvenile arthritis, and some interferonopathies, which are inflammatory diseases characterised by overproduction of type 1 interferons. So on the right of this slide is just some clinical outcomes in rheumatoid arthritis, showing the JAK1/2 inhibitor baricitinib against standard care or placebo. And I'm not going to go into detail of this slide, but hopefully you can see that the patients that received baricitinib had better outcomes than those that received the standard care. So this is these JAK inhibitors are real blockbuster drugs in the rheumatology world. But as with all drugs, there are some side effects of JAK inhibitors, and these include risks, increased risk of infection and some hematological and biochemical abnormalities. So on the right here, we're looking at trials with the JAK inhibitors, tofacitinib and baricitinib, compared with several different studies, including registry data. And what we can see is an increased risk of infection, or more common infection with JAK inhibitors, but no increase in malignancies in these studies that have got very large numbers of patients. Okay, so now I'll just talk to you about the preclinical studies that we've done. So we've approached several different pharmaceutical companies that make JAK inhibitors to try and test a number of different ones in our models. And the data I'm going to present shows a little bit of a mixture of data from using different JAK inhibitors, and where I think I've labelled those on the different slides that so that you can see which one's which. And sometimes I've just put JAK inhibitor, and this means that I guess in all cases with all the inhibitors we've got the results are the same. So as Tom said to you, class 1 upregulation is dependent on interferon gamma, and interferon gamma, as we know, signals through this JAK-STAT pathway. So here we've just blocked upregulation of class 1, HLA class 1 on human islets with the JAK inhibitor. So we've cultured islets with interferon gamma, we see an upregulation of MHC, and that's blocked by JAK inhibitors. And we teed up with Misty Jenkins, and Tom showed you one of the videos that we produced looking at cytotoxicity of beta cells by CD8 positive T cells. And here what we've done is we've cultured our beta cells with vehicle or with the with JAK inhibitor, and looked at the calcium flux and synapse time of the CD8 T cells on the beta cells. And so the video at the top here is the vehicle, and if I play that, you can see the very bright green flash and the long synapse time of the T cell with the beta cell. And in comparison, if I show the bottom video, which is the JAK inhibitor, in this video, it's quite graphic actually. The T cells really swim around the beta cell. They don't make very long synapses. They don't have very bright calcium fluxes. And consistent with this reduced calcium flux and reduced synapse time, we also see reduced cytotoxicity of islet cells by CD8 positive T cells when we use this JAK1, JAK2 inhibitor from AstraZeneca AZD1480. So in addition to reducing MHC class 1, we also see a reduction in inflammatory genes in islets after culture with cytokines. So here we've taken human islets and cultured them with a cytokine cocktail, and with or without the JAK1/2 inhibitor ruxolitinib. We see a reduction, we see an upregulation of some of these genes with cytokines, and this upregulation is prevented by adding the JAK inhibitor. Also, this beta cell phenotype gene PDX1 is downregulated by cytokines, and that's prevented by the JAK inhibitor ruxolitinib. So I guess just to conclude that little section, the JAK inhibitors are able to block the hallmarks of inflammation in the pancreas that are mediated by interferon gamma. But interferon gamma deficient NOD mice develop diabetes, and this has been known for some years now. And we've been interested in studying this along the way. And here's just a diabetes incidence curve that was produced by Tom Brodnicki, who made the interferon gamma receptor knockout mice using CRISPR directly in the NOD genetic background. And we did this partly because there's been some doubt cast over results with hitchhiker genes in the interferon gamma receptor mice that have been backcrossed onto the NOD background. And so we wanted to make these directly on the NOD background. But what you can see here is that the diabetes incidence is almost identical in the interferon gamma receptor knockout mice. So if blocking interferon gamma itself doesn't prevent diabetes, then how else are the JAK inhibitors working? Here's the list of cytokines that are signaled through the JAK-STAT pathway, and as we've talked about interferons already, but we're also interested in the common gamma chain cytokine families. They're interleukin 2, 7, 15, 21, and these are important in lymphocyte proliferation. So we wanted to look at whether our JAK inhibitors are also blocking this family of cytokines in development of diabetes. So first we just looked in vitro at in blood cells. So we've taken some mouse blood here, cultured it with either IL-7 or IL-21, and used the JAK1 selective inhibitor ABT317, this is from AbbVie, and looked at a phosphorylation of STAT5 and STAT3 by these cytokines, and we can see an increase in STAT3 phosphorylation here, which is prevented by the JAK inhibitor, and that's the pooled data is shown over here. When we look in vivo, we can see here what we've done is taken some NOD mice and treated them for 3 days with twice daily oral gavage of vehicle or 2 different doses of our JAK1 selective inhibitor. And then 2 hours after the last dose of inhibitor, we take some blood, stimulate it with IL-21, and then look at phosphostat3 in the T cells, and we can see that both doses of this drug inhibit the phosphorylation of STAT3 in blood cells in vivo in our NOD mice. And these common gamma chain cytokines have been implicated in autoimmune diabetes, and indeed blockade of IL-7 and IL-21 can prevent and reverse diabetes in NOD mice. Then we did some in vivo experiments where we've treated our NOD mice with JAK inhibitors. And so Tom showed this diagram of the time course of diabetes in NOD mice before. So what we've done is taken mice before they become diabetic, but after they've developed an increase in inflammation in the pancreas, which we call insulitis, and given them 2 weeks of treatment with twice daily oral gavage of our JAK inhibitor. Here we've looked at the development of effector memory cells in the spleen, and we can see a reduction in those cells with our JAK1 selective inhibitor, ABT317, and also a reduction in the proportion of KLRG1 positive cells, which is a marker of effector cells. And then similarly, if we look in the islets with the same time course of treatment, we see a reduction in the proportion of CD45 positive immune cells, the number of CD3 positive T cells, and also the number of beta cell antigen specific CD8 positive T cells as measured by tetramer staining. And consistent with this reduction in immune infiltration in the pancreas, we also see an inhibition of upregulation of MHC class 1, which is very similar to a very old data that Tom showed you, where we see reduced MHC on the beta cells in mice that don't have interferon gamma receptors. Okay, so then we tried our JAK inhibitors in a couple of in vivo models of diabetes, and this first one that I'm going to show you is a CD8 dependent model where called NOD8.3. These are TCR transgenic mice that express that have T cells that recognise the islet cell antigen IGRP. These mice develop diabetes a bit more rapidly than NOD mice and with slightly higher penetrance as well. And so when we treated our NOD8.3 mice with a JAK1, JAK2 inhibitor, the AZ compound, we see a significant protection from diabetes. And this is characterised by, you can see in the histology here, a reduction in the inflammatory infiltration of CD8 positive T cells into the islet. So here we've got the insulin staining, and here we've got CD8 staining. In our vehicle treated mice, we have very little insulin staining with a very large number of CD8 positive T cells in the islets. This is in the pancreas, these stains. And in our JAK inhibitor treated mice, we can see quite good preservation of insulin staining and very little inflammatory infiltrate. And I just want to point out that this appearance in the pancreas is quite different to what we see in the same model that overexpress the SOX1 transgene in the beta cells. And Tom showed this before, showing protection from diabetes with overexpression of SOX1. And when we look in the pancreas of these mice, we see these are pancreases that have been stained with insulin. We can see the beta cells here, but they're surrounded by a massive amount of inflammatory infiltration. And so this is sort of shows that the JAK inhibitors not only protect the beta cells from being killed by the CD8 positive T cells, but they also reduce the inflammation in the pancreas. So they're sort of somewhat better than using transgenic expression of RIPSOX in that regard. And then the second model of diabetes that I want to show you is the reversal of newly diagnosed diabetes. So here we wait for the mice to become diabetic, and on the day that they're diagnosed with hypoglycemia, we implant an insulin pellet, and this pellet lasts for about 3 weeks, so returns the mice to normal glycemia for approximately 3 weeks. And during this treatment period of 40 days, we give them oral gavage, twice daily oral gavage of JAK inhibitor or vehicle, and we monitor their blood glucose levels. And so the results are here. This grey box is the treatment period with the JAK inhibitor, and the first 3 weeks or so, the mice remain normoglycemic because they have an insulin pellet. And then after the pellet wears off, you can see the vehicle-treated mice return to hypoglycemia, while the JAK inhibitor-treated mice really remain quite well protected from hypoglycemia for the whole of the treatment period. Now, after the treatment stops, the hypoglycemia does return in these mice, and the higher dose of the drug is slightly better than the lower dose of the drug. So together with the team at AbbVie, we did pharmacokinetics on with their drug, the ABT317 drug, and we can see that a couple of things here, that the plasma concentration of the drug declines quite dramatically over time, which is why we're doing twice daily gavaging. And also the team tell us that this plasma exposure level is similar to what they see in the humans in their trials with upadacitinib, which is, this drug is the sister compound for upadacitinib. Okay, so JAK inhibitors block interferon gamma and common gamma chain cytokines, and so we think that that's how they're working to prevent type 1 diabetes. So as we've said, interferon gamma upregulates class 1 on the beta cell and facilitates the interaction between T cells, CD8 T cells and beta cells, and it also is involved in activation of the CD8 T cell. But the JAK inhibitors can also block expansion of the CD8 T cells and block expansion of the CD8 T cells. And over here, I've just got some data showing islets that have been isolated from NOD mice and cultured in vitro with IL-2. When we do that, we see expansion of autoreactive beta cell specific CD8 cells, which is shown here, and this is completely blocked by the addition of interferon gamma. So just to conclude our preclinical data, we validated the use of JAK inhibitors in type 1 diabetes, showed that they reverse newly diagnosed diabetes, they prevent interferon gamma signaling in both beta cells and T cells, which is something that makes them highly attractive as a immune therapy, and they prevent common gamma chain signaling in T cells. And so the next step then is to progress these inhibitors into clinical trial in type 1 diabetes. So I'm just now going to very briefly introduce you to our trial, which is called BANDIT for baricitinib in new onset type 1 diabetes. And these are the main players in the trial. So we've got Michaela Webble, who's led the project management, John Wentworth, who you all know, who leads the clinical team. We've got Stuart Mannering, who is heading up the mechanistic studies group, and Bala Krishnamurthy, who's a clinician researcher heavily involved in all aspects of the trial. And the trial is funded by the Juvenile Diabetes Research Foundation. So the photo in the middle is one that I found of Tom and I in our little corner of the lab on the 3rd floor. It looks like we're doing an insulin secretion assay here, and it's back in the days when we used to do experiments, when we didn't have grey hair and when Tom wore glasses and I didn't. So yeah, we've been collaborating for a very long time on this work, I guess. So here's a little timeline of what we've been through to develop this trial. So Tom and I first approached Andrew Wilkes, who, as you probably know, was one of the first to identify the JAK molecules back in 2012. And Andrew's the one that suggested we approach several different companies developing JAK inhibitors, which is what we did. We got the AZD compound in 2013 and did our preclinical evaluation before approaching Eli Lilly, who gave us their baricitinib sister compound. We met with the JDRF in 2016 and they introduced us to the team at AbbVie, who gave us their JAK1 selective compound ABT317. We published our paper in Diabetes that year. And then in 2018, in May, Tom and I visited Eli Lilly to talk about the idea of doing a trial. And that set off a sort of long process of us applying for JDRF funding and also investigator-initiated trial approval from Eli Lilly. That came through in 2019, the funding in 2020, earlier this year, and the trial is set to begin this year. So the trial is a double-blind placebo-controlled study with a 2-to-1 ratio of baricitinib to placebo in new onset participants aged 12 to 30. We're going to do treatment for 48 weeks with a further 48 weeks of follow-up. And the primary objective is to assess the preservation of meal-stimulated C-peptide in new onset subjects, and you may remember Tom showed a slide of the decline of C-peptide in new onset subjects, and what we're trying to do is prevent that decline. We've got safety and some other clinical parameters as secondary endpoints, and we have a number of mechanistic outcomes to evaluate diabetes-specific immune responses to our JAK inhibitor. And so the trial was set to start this year, but has been a little held up because of COVID. And I just wanted to finish with one small bit of information about baricitinib and COVID. So there is potential for baricitinib to benefit at multiple stages of the disease. It was identified as an inhibitor of a kinase AAK1, which is downstream of the putative receptor for SARS-CoV-2. And cytokines have been identified as pathogenic mechanisms on severe COVID. So there is a potential for JAK inhibitors to play a role in treating this stage of the disease. But of course, it could also reduce immune responses, including innate immune responses to virus infection. And so these two things have to be balanced. But there are multiple clinical trials of baricitinib in COVID-19, and although there's not much data yet, a small trial in Prato in Italy used baricitinib with antiviral agents and saw benefits of baricitinib. As yet, there's no increased evidence of increased hospitalisation in COVID-infected people who are taking JAK inhibitors for their RA. So we remain cautiously optimistic about using JAK inhibitors in the current times. Okay, and I just want to finish with the acknowledgements. So the main people that I want to point out are Prerak Trivedi and Ting Ting Ge, who did most of the who produced most of the data that I showed, along with many other lab members, and I've listed some of the main people from the past here. But there are many people who've contributed to this work, which is actually a couple of decades. I also want to acknowledge Stacey, who is our animal technician who works in our laboratory. She does all of the animal work. She also isolates the islets and does all of the does a lot of the in vitro work as well. And she's done an amazing job at coordinating all the twice daily gavaging of our mice. We've had some really nice collaborations with some people at WeHi, including Misty, Chris Burns, Andrew Liu and Robin Sutherland, who we continue to collaborate with. And also our collaborations with Mario Ehlers at Eli Lilly and Lisa Olsen and Andy Long at AbbVie, who've helped us really understand how these drugs are working. And then finally, I just want to thank, I've already mentioned the people in the BANDIT trial, but I want to thank all of the admin people who have helped with getting the trial up and running, and the funding bodies. And yeah, thanks very much.
That's terrific. Thanks very much, Helen and Tom. Now, I can't see any banter on the live Q&A. I'm not sure if I'm reading the right thing or not. I'm not sure if you can comment, Colin, as to whether I'm missing questions, but I might just throw one in as the chairman's prerogative. You know, in light of the fact that the interferon gamma receptor knockouts are not protected from diabetes, do you think that the mechanism is going to be primarily one that's mediated through immune cells rather than through beta cells and preventing their upregulation of class 1 MHC?
Well,
do you want me to take that one?
Well, maybe we'll both have a crack. I'd say, I mean, my view is that we went through a phase of really thinking we would like to block the interaction between immune cells and the beta cell, because and mainly focusing on the beta cell. But I think what we've come around to is that treating both is the right approach. There are complex reasons why the interferon gamma receptor deficient mice still develop diabetes, but certainly the idea that we're blocking both the gamma common chain pathway and the interferon gamma signaling pathway, that to me is why the drugs are so potent, and they wouldn't be as potent if they did one or the other.
Yeah, I agree with Tom. I was going to say that too. And it's definitely clear from our preclinical work that they are doing both. You know, they're having effects on the T cells and they're having effects on the beta cells as well.
And look, a second question I had, again, I'm not sure if I can or can't see any questions being put in from the wider audience, I'm sorry. But
there's a couple here.
Are there? Well, do you want to take them on then? Tell me what they say.
Under the published column, John, you can see them here. No?
No, you take it away. I'm inept, I'm sorry.
Okay, so someone anonymous says, very nice talk. I'm wondering whether there are any known genetic environmental factors promoting or affecting infiltration and interferon production by those T cells, which can lead to class 1 upregulation and further apoptosis in beta cells and type 1 diabetes patients. Yes, I guess one of the there's some evidence that virus infections are involved in triggering diabetes, important environmental factors, there's a lot of data out there indicating an increase in type 1 interferon genes in islets and in people before they develop symptoms of disease. So this would be certainly one factor that could result in upregulation of class 1. I mean, we do see upregulation of class 1 by type 1 interferons, but not that's not important. If we block that, it's not important in disease progression in not mice for sure.
Do you want me to respond to the next question, whether baricitinib can bind to the class 1 peptide binding groove just like abacavir and hence interfere with peptide binding to present to T cells? Well, look, to my knowledge, it cannot, but there is a current trial of a repurposed drug, alpha-methyl-dopa, or Aldamet, an antihypertensive drug that is being used for exactly that reason as a drug which can interfere with, I think, peptide binding to the class 2 molecule rather than class 1. So that is a mechanism of drug action that is of interest in diabetes. We've not. The second part of the question was whether we've treated momelotinib, which is another JAK inhibitor, which we haven't actually. One of the things that we've really tried to do is to use a whole variety of JAK inhibitors, and this was a piece of advice from the very wise Andrew Wilkes, who of course discovered the JAK kinases, who suggested that having a portfolio of drugs that you test is a powerful way to influence the pharmaceutical industry, because you can, you come along with a portfolio that you can then, you're not left with just one company to approach. But we've not actually used momelotinib, but we are interested. There's a very big family of drugs now, and we are interested in testing other JAK inhibitors.
Now, thanks, Tom. Look, I've found the questions, so thanks for Colin for publishing them. The one is, how long is the trial expected to take? Do you want to take that one on?
Well, maybe you can take that one on, John. Yeah, so the enrolment period is expected to be 2 years, and the follow-up period will then take a further 2 years. So altogether, we've got 4 years of funding.
Yeah, so it's a long process. And then an anonymous question, is it just one antigen presented by MHC in type 1 diabetes?
No, it's not. So just like the autoantibodies, which recognise multiple different antigens, there's a polyclonal T cell response that not only recognises one antigen but recognises several. They're different but overlapping in the mouse and human model. We believe in our group that insulin itself is a very important antigen, particularly for CD4 positive T cells, but also for CD8 positive T cells. But there are several antigens which are known to be recognised by CD8 positive T cells in diabetes.
Now, I can't see any additional questions, but I might just throw one final one in there, if I may. And, the way we think we're going to have to proceed in this field is much the same way that we've done in transplant rejection and autoimmunity, and that is we need to combine agents that are effective to get synergies. Could you comment on what you might want to stick with a JAK inhibitor if it shows that it's effective in type 1 diabetes? Have you thought about what might work in combination with it to synergize to really fully protect beta cells and provide durable prevention, perhaps for only a limited treatment window?
Yeah, well, one of the things that our group's quite actively working on is combinations of antigen-specific therapy with JAK inhibitors. And we have a little bit of data to suggest that if we use antigen-specific therapy on its own, it's difficult to block the antigen-experienced cells. But if we use it with JAK inhibitors, so the JAK inhibitors will eliminate many of the cells, and then we can use the antigen-specific therapy and get more longer-lasting protection that way. So that's something that we're very interested in exploring further.
And that protection is through regulation, Helen, I presume, enhancing immune regulation, or not sure.
Yeah.
Multiple pathways,
Yeah.
multiple mechanisms.
Yeah, that's right. It depends a little bit on how you induce the tolerance, I think.
Yeah, I mean, we're very interested, for example, in T-cell exhaustion, which is a concept that derives from immuno-oncology. But as Helen says, it would be great if you, and you alluded to also, it'd be great to work out a therapy that meant that people with diabetes or with at risk of developing diabetes didn't have to take lifelong therapy. It would be great to work out something that meant that therapy could be intermittent, or even ideally in the end could be a single course of therapy. And while we're talking about therapy, I just want to apologise for my appearance. I've been making the most of working from home to deal with my solar keratoses. So don't worry, everybody, it's self-inflicted.
It doesn't seem like it's not
iatrogenic.
It's not a recognised COVID symptom or sign, so you don't necessarily need to stay at home. Look, thank you very much, Helen and Tom. We've come to the top of the hour. Really appreciate you joining us today, and I hope everyone has enjoyed that, and we'll catch up in due course. Thanks very much.
Thanks a lot.
Thank you.